Chapter 10, How Cells Divide, also called the Mitosis chapter. The cell cycle begins immediately after the previous cell division. This begins in two periods, interphase and mitosis. Mitosis is the portion where the cell actually divides. Interphase is the longest part of the cell. During interphase, the cells grow, carry out their normal functions, and prepare for mitosis. Interphase is made up of three phases. The G1 phase, which is called gap 1. This is because this is the time between when mitosis ended and the DNA synthesis for the next cycle will begin. The synthesis phase, which is when DNA will be replicated, and the gap 2 phase. This is the time between synthesis and the actual start of mitosis. In the G1 phase, the cell performs its normal functions. It begins just after the new cell is formed, so the cell also is growing during this time. This is usually the largest period of time in interphase because it's when the cell is doing its job. At the end of the G1 phase, the cell will rest before going on to DNA synthesis. The rest phase is called the G0 phase. After the resting phase, the cell begins synthesis, which is S. This is the most important part of the interphase cycle because this is when DNA is copied. This is the DNA that will be used in the new cell. We will talk about this in the DNA replication chapter, which will come in a few weeks. In G2, cell growth is continuing, but more importantly, the organelles are being duplicated at this time. The microtubules also begin to reorganize and form spindles. Spindles are going to be used to hold and to move the chromosomes during mitosis. Remember, DNA, when it's being used, is in long, thin strands called chromatin. When they are all condensed and thickened, that's when they become chromosomes. In this picture, we're just showing you the rest of the cycle. See how G1 is very large, takes up most of the interphase. Then there's the synthesis, which is the red section, and the green G2. Mitosis is shown in purple, those individual steps we will talk about, and then cytokinesis, which is where the cell actually splits into two cells, is the blue arrow. The amount of time that a cell spends in interphase or in the cell cycle depends on the organism that you're talking about. Embryonic cells, those cells that belong to embryos, can go through an entire cycle in 20 minutes. That's because they don't actually grow. All they do is duplicate as fast as they can copy the DNA. A fruit fly can go through the entire cycle in 8 minutes. Most mature cells will take longer. So, for example, an animal cell will take about 24 hours to go through this entire process. And this is just a picture of what the cell looks like during interphase. Notice you have the nucleolus is still there, the nuclear membrane. The green parts are things called asters. The middle part is the centriole. The centriole is where the spindle fibers are going to come from. And those little green lines are the actual spindle fibers. The centrioles plus the spindle fibers together are called asters. Mitosis is divided into these 5 phases, and we will be going through them step by step. Prophase is the first step. The chromatin are going to thicken and coil and turn into a thing called the chromosome. When the chromosome is condensed, it's easier to move, but it cannot do its job. It can't be duplicated, and so it's inactive in this form. The nuclear membrane and the nucleolus break down. The centrioles begin to move to the opposite sides of the cell. As they move, they create those spindle fibers that I talked about in the previous slide, and then they create those asters. Biologists aren't really sure what the asters do, but they believe that they provide strength for the spindle fibers. When the centrioles have finished moving, you'll have an aster at either side of the cell. The spindle fibers will stretch between them, and they will create what is called the spindle apparatus. Prometaphase is the transition period between prophase and metaphase. This begins after the nuclear membrane disappears. During this time, a second group of microtubules will begin to grow from the asters, or from the poles, and will attach to the chromosomes at the centromere. The centromere is the part that attaches the original chromosome with its copy. This is important because that connects each chromosome to each centriole, and they will be used to move the chromosomes around as the cell gets organized, as it prepares for duplication and for cytokinesis. The microtubules begin to move the chromosomes toward the center of the cell. This movement is called congression. The movement is jerky, meaning that it pushes the chromosomes toward the middle, and then they pull it back and they adjust it back and forth until it's completely lined up. The next phase is metaphase. This is where the chromosomes are tweaked into place. The spindle fibers finish that movement until everything is directly lined up along the exact center. This center is called the metaphase plate. If the chromosomes are aligned correctly, the centriole will have moved each chromosome so that the centromere is exactly on the metaphase plate. This is going to be important because if you look at the picture on your right, you can see the metaphase plate, and that will divide the chromosome from its duplicate in the next step. Anaphase is the shortest of all the phases. This is simply because there isn't as much going on. It begins when the centromeres split, creating two separate chromosomes called sister chromatid. This happens when the proteins holding the chromosomes together are broken down. Those proteins are called cohesion proteins. The spindle fibers begin to shrink and become disassembled, and as they do that, they get shorter. As they get shorter, the chromatid are pulled toward the poles. So you'll have the original chromosome pulled to, say, the left side, and its duplicate pulled to the right side. This happens in two steps. Anaphase A, where the spindle fibers become shorter so that the chromatid are actually physically moved, and anaphase B, where the poles begin to move apart. This again makes the chromosomes move away from each other while the cell becomes more oval in shape, almost like a peanut. Telophase is the reverse of prophase. Everything that happens in prophase is removed. So the spindle apparatus is disassembled. The parts will be used later to create the new cytoskeleton in the new cells. The nuclear envelope reforms. The chromosomes begin to uncoil and go back to becoming chromatin. The nucleolus is going to reappear, And then the organelles are divided up. Remember, they were copied in G2, so that when everything is done at the end of telophase, you have one of everything all kind of crammed into one cell. So it's very crowded because the two cells are still connected. Then you have cytokinesis. Cytokinesis is the actual separating of the cell. In animal cells, which is the top picture, this happens as the proteins, they're called protein filaments or actins, begin to constrict. And they get smaller and smaller and smaller, pinching and pinching and pinching until the two cells actually come apart. In a plant cell, the cell plate forms between the two nucleus or the nuclei, and it builds until it gets thick enough that it fuses to the plasma membrane, and then the cells are separated. This is a picture of all of the phases. You can follow from arrow to arrow to arrow and see how the chromosomes are moved. In mitosis, its job is to help the cell grow, or to help the organism grow, to help it to become developed and change, and to repair cells that have been injured or cells that are old. In order for them to become different, because all cells immediately after mitosis are exactly the same, they have to go through the process of differentiation, meaning taking on new shapes or taking on new jobs. That happens when a chemical called a growth factor is created, and the receptor proteins on the outside of the new membrane receive them, and that growth factor says, you are a heart cell, start behaving like one. There are 3 checkpoints in the cell cycle where the cell goes back and checks its work. The first one is the G1S checkpoint. This is the beginning where the cell decides whether or not it's going to divide. It looks for, is there enough food? Is there enough information? Meaning, is the DNA okay? Is it damaged? Is there a growth hormone that says it's time? Those sorts of things. If everything is good, the cell continues. Once the cell passes this checkpoint, it must divide. The G2M or checkpoint, this is when the cell makes the commitment to mitosis. It checks to make sure that the DNA has been properly replicated. If the DNA is okay, then mitosis begins. Once mitosis begins, it must be continued. If an error has been made, then the cell stops and apoptosis happens. The last cycle is the late metaphase. This is where it checks to make sure that all the chromosomes are attached to the spindles. They're all lined up properly. If everything is exactly the way it's supposed to be, then the cell will move on. But once it is moved to anaphase, it can't stop. If any point that a mistake has been made and it doesn't pass the checkpoint, apoptosis takes place. That, remember, we talked about is programmed cell death. Occasionally, the cell cycle gets lost. When that happens, the cells begin to grow uncontrollably. You have tumors, which are just cells that are out of shape, cells that don't do their job properly. If they're just poorly shaped, they are called benign tumors. Those can be easily removed. If they are malignant, you have cancer. These cells are abnormal in their size, abnormal in their shape, and they have ability to take normal, healthy tissues and turn them into cancer cells, causing the cancer to spread. This is the end of Chapter 10, Mitosis.